Hey, how's it going? Welcome back from break. Today we're going to be talking about gene expression, and mainly the goal is for today is to find out how is DNA the instructions for life. So to understand why DNA is the instructions for life, you have to start off with the idea of the central dogma. So this is just going to be a brief outline of what we're going to cover today. Today we're going to talk about the central dogma, which is the idea that DNA, which again is a double-stranded helix. You might remember this from macromolecules, that there are 4 macromolecules, including carbohydrates, proteins, DNA, and our last one is lipids. So we go from DNA, we're going to something called messenger RNA. Again, this is another one of the nucleic acids. And lastly, we're going to get to proteins. And how DNA is the instructions for life is because of this idea of what proteins can make. So proteins make you who you are. You're made of proteins. And they make decisions about how you're going to act, how you're going to behave, what you're going to do, what your hair color is, what your eye color is going to look like. And who you are is your phenotype. So today what we're going to cover in this lecture is how do we replicate DNA, how do we make more DNA. So that's called replication or DNA synthesis. We'll talk about how the DNA message gets turned into messenger RNA, which is called transcription. And lastly, we'll talk about how messenger RNA gets made into protein, which is called translation. So what we'll cover today is this idea. How do we get from DNA to protein that makes you who you are? First up, we'll focus on DNA. Remember, DNA is something that's double-stranded, and it's found in the nucleus. It's the instructions for life. The big points about DNA is that it's double-stranded, it's in the nucleus. We have 4 bases, 4 things, 1, 2, 3, 4, make up DNA. So these are going to be our things like adenine, which gets paired with thymine, cytosine, which gets paired with guanine. Now within these, there are some characteristics that you might want to remember. Adenine and thymine. Adenine and thymine always pair together because they have a base pair. They're best friends, just like cytosine and guanine. They're best friends and get paired up. So what makes more DNA? If you might recall, DNA looks like a double helix. So there's some steps that are involved in trying to get that double helix to make 2 copies. So we end up with 2 of these. And so who are some of the key players in this? There are kind of 3 enzymes you want to think about. Helicase. Then we have a second enzyme, which is called DNA polymerase. And we have a third enzyme that I want you to remember about called ligase. So first of all, you might want to say, what's these ACEs? I don't know what you're talking about. ACEs are part of a group of things called enzymes. Enzymes have jobs, and in this case, their jobs is to build DNA. So we'll step back and we'll talk about what each of these 3 things do, and then we'll get into transcription and translation. Here are 3 enzymes from before: helicase, polymerase, ligase. What do we want them to do? So helicase, what does it do? It's going to unwind DNA. Remember, it's a double strand. So you've got to unwind it, and so you'll have your typical ladder formation that most people think of when they think of DNA. What does polymerase do? This is poly, many. Remember, poly means many, so we're going to build many DNAs. So we've separated them. We have our old strands, and we're going to build with DNA polymerase our new strand. Lastly, we'll have ligase that kind of glues everything together. Ligase means to glue or put together. So that's how we go about making new DNA. We know what one side is. If one side is adenine, we know that it must always be paired with what? Take a second and think about it. It's a T. Anytime you see an A on this old strand, it'll become a T in the new strand. Anytime you see a C, it'll become a G, and vice versa. That way you can use the old strand to make the new. So ta-da! DNA replication. Take a moment and summarize. Can you tell me how to make DNA? Why is it important? If you can, go ahead and go on to the next step. Otherwise, flip back, reference your book, see if you can answer that question to how DNA is made and why it's important. We're going to talk about transcription. Transcription is the idea that you're making a message. So it's really important that you keep DNA locked away inside your nucleus. You don't want it ever leaving. So what can you do that can leave instead? Messenger RNA. Remember, message, something you send. It's like sending an email. Instead of sending around your social security number or maybe your birth certificate or something really important to you, you can kind of make a copy, a message that sends the exact same information. So we'll send a message using mRNA. Use the same rules as before, with the only difference being instead of a T, there's no T in transcription. So instead we'll use something called uracil, which is our U. So anytime you see an A, it'll go with a U. Anytime you see a G, it'll go with a C, and vice versa. So that's how we get to transcription, which will make a messenger RNA. This is our message that's going to go out to the cytoplasm. So this guy's going to go outside the cell, or outside the nucleus, rather, outside to the cytoplasm. So we're outside to the cytoplasm, and where will we go next? Now we've transcribed it. We have a message. This message can then go out to the cytoplasm. When that message is outside, it's taken up by something called a ribosome. This is the machinery that reads it, that quote-unquote reads the mRNA. And how it reads it is by bringing in these tRNAs that carry amino acids. So what do amino acids make up? Think back to last semester. Take a moment and think back. What do amino acids make up? What is it the monomer of? Proteins! Woohoo! So amino acids make up proteins. And so through translation, because of what is on this mRNA, it tells you exactly what's going to go on this side. So for example, a very popular one, AUG. What goes with A? U. What goes with U? A. What goes with G? C. UAC codes for a very specific amino acid, and that one is methionine. So anytime you would have an AUG on the mRNA, it must match up with a UAC that would allow you to put a methionine. Over time, these amino acids can bond together using something called a peptide bond, and as they bond together, you're going to get a growing chain. Eventually, it'll lead to a stop codon, just like when you're driving. You will come to a stop sign telling you to stop translation, and that'll lead you to making a protein. So that's kind of the two steps, transcription and translation, really quickly. Let's go back and decide. What's different? Compare and contrast transcription and translation. What do they have in common? What do they have different? That'll be kind of your first step to make sure you understand these ideas. Take a moment, compare and contrast translation and transcription. What do they have in common? What do they have different? What are the big players? Hopefully you've taken a minute to compare and contrast DNA transcription and translation. You remember why DNA is important. So to summarize, let's kind of hit all those big points again on why all this stuff is important. So again, DNA is important because DNA acts as the instructions for life. It tells you who you're going to be in some ways because it's the recipe for making proteins. That's step one. You have to have DNA, you have to have the recipe for making proteins. Once you have the recipe, then you can do things like transcription. You're going to write the message. So using DNA, you can make a message. That message is then free to float out of the nucleus, again, to the cytoplasm. And lastly, we've got translation. And translation, again, is important because you're finally putting all the pieces together. Just like when you're making a cake, you're finally building your protein. And you're going to be using things like the messenger RNA, tRNA, which carries your amino acids, and the ribosomes. So this is how you become who you are, because your body has instructions that tell you kind of who you're going to be, what you're going to look like, because of transcription and translation. So that's kind of the breakdown for this week. Hopefully it's something helpful. You can think about DNA, transcription, and translation. Remember to make sure you practice. The really important part of this skill is seeing it, being able to do it, being able to remember the base pair rules. So that's the game plan for this week. I hope you had a great break, and I will see you next week when we talk about things like mitosis and meiosis. Take care. Bye.